Hope so. Great. Yeah, really sorry I couldn't be there in person, that the flights didn't work out, but I've been asked to speak today about the role of latent viruses as drivers of chronic illness states. So as was mentioned previously, our research program in San Francisco is called LINC, which stands for Long-Term Impact of Infection with Novel Coronavirus. And this study launched in March of 2020 to try to understand what happened to people after they got COVID, based on the assumption that it would be more complicated than a 2-week illness, which unfortunately turned out to be true. So we've been in operation through all of the waves of the pandemic. We've enrolled over 800 participants so far. We see everybody 3 to 4 times a year. We've banked tens of thousands of biospecimens and have supported dozens of collaborations focused on understanding the mechanisms of long COVID. And for the last year and a half, we've been really fortunate to be a part of the NIH's Recover initiative and have co-enrolled many of our LINC participants into the Recover program and are active in that program. So there are many proposed mechanisms of long COVID. Most of these will be discussed at some point during this meeting, and I've been asked to speak specifically to viral reactivation in long COVID based on some work that we published earlier this year. So most viruses, most infections come and go, but some infectious pathogens can establish latency or persist. And in terms of viruses, we think about this most commonly with herpes viruses, which cause things like cold sores, shingles. Other herpes viruses like EBV and CMV can cause systemic illness, particularly in people who are immunocompromised. And then there are a whole variety of other pathogens that can establish latency that are sort of beyond the scope of this talk. I'm really going to focus in my comments today on EBV and CMV and show you some data from long COVID that I think will inform the role of these viruses and other infection-associated chronic illnesses. So I'll start with Epstein-Barr virus, which is a quite ubiquitous herpes virus that almost everybody in the developed world ends up contracting at some point. EBV infects B cells and resides in memory B cells indefinitely. The initial infection leads to the classic mono-like syndrome, but people can also have very mild or even asymptomatic EBV infections. And it was initially identified in association with both hematologic malignancies like lymphomas, leukemias, and epithelial malignancies. The biology of EBV is really interesting. So once a person is infected, it establishes latency in the form of a circular episome, genetic material that is tethered to human chromatin by a protein called EBNA. And EBNA is important because we can actually measure the antibody response to that protein as sort of a marker of disease activity. In the latent phase, there's minimal gene expression, and EBV relies on normal cellular division to passively move on to various daughter cells. But under certain conditions, EBV can reactivate and enter a lytic cycle. So in the lytic cycle, the virus wakes up, it expresses all of its viral genes. This results in cell death, production of viral progeny, infection of other target cells. And this can be inhibited by antivirals like acyclovir or acyclovir. But an important point to note is that clinically, typically the treatment for EBV reactivation is to intervene on whatever other underlying condition is leading to the ability of EBV to sort of reactivate. We only rarely directly treat EBV in most clinical contexts. The immune response to EBV is very complex, and I'm not going to go into it in full detail, but I think the figure on this slide is important because we can measure the serologic response to different components of the EBV virus, and that gives us a hint as to what's going on in terms of the viral activity. So during an initial infection with EBV, there are certain antibodies that come and go, the viral capsid antigen antibodies. And then there are other antibodies that persist for different amounts of time. So A person with EBV infection typically has high-level VCA responses forever, not really useful in a clinical context. There are responses to early antigen, which peak around 2 to 3 months after initial infection, and then in the vast majority of people wane to undetectable levels after about 6 months. And so even though somebody still has the infection, that antibody typically goes away. And then there are nuclear antigen or EBNA antibodies that peak later and persist at kind of moderate levels and may be a good metric of kind of the total burden of EBV in a person's body. As has been mentioned previously, EBV has been implicated in a variety of other conditions, including autoimmune diseases, but these are almost always associations rather than clear causal relationships. And so there are associations that have been identified between EBV and lupus and rheumatoid arthritis. And there's a lot of work that's ongoing now to really understand the role of EBV in these conditions. I think one of the most exciting discoveries of the last few years was the potential causal role of EBV in multiple sclerosis. And I think there's going to be a dedicated session at this meeting on that topic. But this study was a study of 10 million people in the US Armed Forces, where they basically found that, that people who seroconverted became EBV positive after joining the military had a 32 times risk of developing MS subsequently. There are various mechanisms that have been proposed for why this might be. This includes molecular mimicry, so immune responses against EBV kind of cross-react against human proteins and cause trouble, as well as older autopsy studies showing direct infiltration of EBV-infected B cells and plasma cells into the brains of people with MS. This, isn't really EBV reactivation in the sense that I'm talking about it, but really showing that EBV infection can make certain people vulnerable to developing MS. There have also been a good number of studies of EBV in chronic fatigue syndrome, and I think there will also be a session on this topic. Herpes viruses in general have commonly been associated with ME, and this includes both the presence of viral genome sequences that can actually be detected, suggesting kind of a lytic phase, or elevated antibody titers against EBV. But my assessment of the literature in ME/CFS is that it's quite heterogeneous and quite mixed. And so it's difficult to understand what's actually going on, whether EBV is driving ME or whether kind of the stress response to ME and the immunologic response to ME is creating an environment in which EBV can reactivate. And so I think more work really needs to be done to understand this in ME/CFS. There are a few really provocative studies looking at antiviral treatment against EBV in ME/CFS. These are mostly studies of valganciclovir. They're typically small studies, 10 to 50 or so people, often single arm, occasionally randomized. But it seems that in some people, particularly those people who might have high level EBV burden in their body, actually have a benefit from this type of treatment. And so while this hasn't been studied at scale, as far as I'm aware, I think that a signal may exist here, and I'm hoping that this can be pursued further. I will also say that, a large number of people with ME have trialed valganciclovir, and some have reported improvement. So I think this was a natural thing to study in long COVID. And the first study that looked at this that I'm aware of was this one, which was shown earlier by Dr. Henrich, a small study that showed that 20 out of 30 participants with long COVID had serologic evidence of EBV reactivation. And this was a much higher proportion compared to people who had had COVID and fully recovered. And really interestingly in the study, they almost saw a dose effect where the number of reported long COVID symptoms had a direct relationship with the magnitude of the EBV titer. So really interesting and provocative observation. This study from the Seattle group in Cell last year showed that EBV detected during acute COVID-19, typically in people who were really sick enough to be hospitalized, was associated with later development of long COVID symptoms, even though people with long COVID did not have evidence of ongoing EBV shedding. So based on those two studies, we decided to explore this in LINK, and this is work that I did with Tim Henrich and Peter Hunt. We studied 280 people in LINK, all of whom had confirmed SARS-CoV-2 infection. These were mostly people who were pre-vaccine through the Delta wave, mostly outpatients. There was a decent prevalence of various comorbidities in this cohort, but overall, these people were pretty healthy, I think in comparison to some of the other studies. It was half men, half women, and we looked at people about 4 months post-COVID. We defined long COVID as any post-COVID symptom that did not predate COVID, so it had to be attributed to long COVID. And we also looked at specific symptomatic phenotypes of long COVID, including fatigue, neurologic symptoms, cardiac symptoms, and GI symptoms. And so I showed the left side of this figure before, but the right side of the figure demonstrates our approach to kind of reading the tea leaves of EBV serology. And so for this study, we looked at early antigen IgG, because in most people with EBV, that should go to negative. And so we looked at people who had positive versus negative early antigen IgG titers. And then we also looked at high-level EBNA titers as sort of a marker of total body EBV burden. We found that about a third to half of participants in our cohort had evidence of one or both of these, but it was very rare to identify people who had circulating EBV in blood. So what did we find? First, we found that those with serologic evidence suggesting recent EBV reactivation, early antigen D IgG, had 2.5 times the odds of reporting long COVID fatigue. We found that those with serologic evidence of high-level EBV responses, EBNA antibodies greater than assay, had twice the odds of long COVID neurologic symptoms like brain fog. We didn't see clear relationships with cardiopulmonary or GI symptoms. And this was all in models that were adjusted for other potential confounders that could be important. We did a follow-up study that was published recently in which we looked at people with cardiopulmonary symptoms and performed cardiopulmonary exercise tests. And we found a really quite striking association between evidence of recent EBV reactivation and cardiopulmonary symptoms, as well as objective measures of things like chronotropic incompetence and lower heart rate reserve in people with long COVID who had abnormal CPET. So I think this is one of the first times that EBV reactivation has actually been tied to abnormal CPET measures. I'm going to shift now and talk about CMV, which is another herpes virus that affects about 60% of adults in the US. A large proportion of our normal immune response is targeted to keep CMV at bay. Like EBV, it can cause a mono-like illness, but most cases are actually mildly or asymptomatic. We typically think about this reactivating in really profoundly immunocompromised hosts, but even in healthy people, CMV seropositivity is generally not thought to be a good thing because it can have lots of immunologic consequences. Earlier this year at CROI, we reported a really interesting observation that people who were CMV seropositive and got COVID were twice as likely to be hospitalized, to get really sick. And among those people who had CMV seropositivity, they had much higher levels of circulating SARS-CoV-2 nucleocapsid antigen in blood. So during acute COVID, CMV seems to be bad. The work from the Seattle group also found that greater CMV response was present in people with COVID compared to people who were healthy, and that increased cellular responses to CMV were associated with GI symptoms 2 to 3 months post-COVID. So we looked at CMV in the study as well, and what we found was really quite surprising. We were expecting that CMV would have an adverse impact on long COVID, that people who were CMV seropositive would be more likely to have long COVID, but we actually found the opposite, that CMV seropositivity was associated with a decreased odds of long COVID, and this held up across all of the long COVID phenotypes that we looked at. So really surprising finding and made us think, why would this be? So there are a handful of papers suggesting that in certain cases, CMV seropositivity could actually be of benefit. This paper was published last year in Science Translational Medicine and showed that young, healthy people who were CMV seropositive actually had really enhanced immune responses to influenza vaccine, and mounted better cellular and humoral immune responses to vaccination. And then, the MS connection, I think, is natural to make with EBV. So what about CMV and MS? There's actually a recent paper showing that CMV may be protective against developing MS. This was a Swedish cohort showing that CMV seropositivity was associated with a 30% decreased odds of MS as opposed to EBV and HHV6, which both increased the odds of MS. So why would these viruses have different effects? Well, one possibility is that EBV acts according to the inflammatory and autoimmune pathways that were just discussed by the prior speaker. It could be related to the fact that EBV and CMV are compartmentalized differently within the immune system, and this could relate to different roles of B cells with EBV and myeloid cells with CMV. And it could also be possible that CMV exerts immunoregulatory effects. For example, it releases a viral IL-10 that can alter some of the perturbations that would otherwise drive long COVID. So one way to potentially put this together is that acute COVID causes an inflammatory response, which results in EBV reactivation, causes autoreactivity, as well as sort of a vicious cycle of inflammation and more EBV reactivation. And ultimately, through some of the pathways that were mentioned before, this leads to long COVID, whereas CMV, although it increases the magnitude of acute COVID, is able to dampen these inflammatory effects and ultimately results in a lower likelihood of long COVID. You can imagine further superimposing some of the other mechanisms like SARS-CoV-2 persistence onto this model. So what are our next steps? We're now doing a study of outpatients during the acute phase of COVID-19 in collaboration with the CDC-funded Find COVID cohort at UCSF. All of those participants are measured at home during acute COVID and then follow up long-term in LINC. We're exploring tissue studies to look at EBV and CMV, for example, in gut tissue. We're working to try to link EBV responses with autoimmunity. And we're thinking about whether antiviral treatment targeting EBV could be of benefit. This could be treatment during the acute phase of COVID, which would be quite challenging. And I think there would be a lot of debate over the relative benefit of EBV treatment as opposed to SARS-CoV-2 antiviral treatment. I think the jury's out on whether treatment during the post-acute phase would be of benefit. It's not clear that there's ongoing viral replication, and we really need to look in tissues to understand that. I think we also would need to think of the collateral effects of EBV treatment on CMV, which is potentially, as I showed you, beneficial. And I think ultimately my assessment of this is all best explored in studies that are large and are kind of happening in big networks. I think there's also an open question of whether other types of EBV targeted treatment, such as cellular therapies to clear EBV infected cells, could be more effective than antiviral treatment because they would target basically the latent phase of EBV rather than the lytic phase. So last slide, in summary, latent viral infections interact in complex ways in those who get sick with other infections. We really don't understand this well. There's growing evidence that these infections associate with long COVID. I think, as Tim mentioned, physicians have really been trained to be skeptical about the role of these viruses. That's definitely true of my training. But I think we need to now take a step back and re-examine and try to overcome this skepticism. In order to do that, I think there needs to be specific prioritization of asking and answering these questions, as well as funding to explore this. And, I think that getting answers about these questions in long COVID may really help to determine the role that these viruses play in the other infection-associated chronic illnesses that are being discussed at this meeting. So I'll end there with some acknowledgements and look forward to the Q&A. Thanks so much.
Thank you.